Hello, curious molecules here. Welcome back to this channel. If you're new here, you're very much welcome. If you're a returning viewer, because most of you view and are not subscribed, please do subscribe. You are also very much welcome. Today we are going to be talking about the TCA cycle. If you are new, you should go and watch the previous videos. We started with carbohydrates and we are continuing with metabolic pathways that involve carbohydrates. So the TCA cycle, tricarboxylic acid cycle, from the name tricarboxylic acid, which means that the molecule that is central here is a tricarboxylic acid. Carboxylic acid or a molecule that is made up of 3 carboxylic acid groups. And it's also called the Krebs cycle, named after the person who found it. And hopefully at least one of you wants a cycle or a molecule to be named after them. As for me, I do. So you have the Krebs cycle or citric acid cycle, because the tricarboxylic acid is also called citric acid, as we'll find out. As with every metabolic pathway or every mechanism in the cell in biochemistry, you want to know why the body has to go through that cycle, for example, why it has to perform the particular metabolic pathway. So you also ask, like, what is the starting molecule? Where does this particular metabolism or mechanism occur in the cell? So for the TCA cycle, it is a common final metabolic pathway. Final meaning that most of the macromolecules, the lipids, carbohydrates, whatever mechanism or metabolic activity they carry out, most of them will end up into producing intermediates that go into the TCA cycle, or producing the molecule that is central molecule in the TCA cycle, or a starting molecule in the TCA cycle. So as we are going to see, the starting molecule in this TCA cycle is acetyl-CoA, which is a 2-carbon molecule that can be gotten from breakdown of lipids, can be gotten from breakdown of amino acids, and also can be gotten from breakdown of carbohydrates, which we are currently seeing. So the TCA cycle, where does it occur in the cell? In the mitochondria. So you can infer here that the cells that do not have mitochondria, like mature red blood cells, do not perform this particular cycle. The major aim of this cycle is also for energy production, but not mostly direct energy like ATP that we saw in glycolysis. It produces intermediates that have to go into the major energy producing machine that we're going to see, which is the electron transport chain. So you have that. And as we mentioned before, to start the cycle, the cell must have acetyl-CoA, which is gotten from other metabolic pathways. And also, in addition to acetyl-CoA, oxaloacetate is also a starting molecule. So these two combine to produce the molecule that starts this particular cycle. So we said that acetyl-CoA is gotten from other metabolic macromolecules, like lipids, like carbohydrates, like proteins or amino acids. So we are currently looking at carbohydrates, and we have seen the most important pathway, which is glycolysis. After glycolysis, the molecule we produce is pyruvate. So how come they're saying that we get acetyl-CoA from carbohydrates? So pyruvate can be converted to acetyl-CoA in the presence of oxygen. And in the absence of oxygen, what does it produce? You remember from the previous classes? So it produces lactic acid when there is no oxygen. But in the presence of oxygen, pyruvate will be converted to acetyl-CoA, and is actually a very important step. It is catalyzed by pyruvate dehydrogenase. Pyruvate dehydrogenase is a complex enzyme. Well, you can say biochemistry is already so complex, so it's not surprising. But this enzyme, when an enzyme is complex, it means that when something is complex, basically, it means that you have to be very careful with it too. So any distortion, any disruption will be very problematic. So pyruvate dehydrogenase has 3 subunits, 3 catalytic subunits. We already know that enzymes are catalysts. They help to speed up the rate of reaction. But they have catalytic sites, that is where the substrate will bind, or the interactions that have to happen. And pyruvate dehydrogenase is made up of 3 of those subunits. So it's like 3 in 1 that are combined together. And all of them have to be together to make the enzyme work. In addition to the 3 subunits, it also has other coenzymes. If you can remember, well, we have talked about coenzymes in one of our videos. And coenzymes are molecules, like when you are co to somebody, that person you guys work together for something to happen. You're a co-CEO, you guys formed an organization together. For it to move forward, you guys need to agree to move forward. So coenzymes need to agree with enzymes. You have to be there with the enzymes to be able to move the reaction forward. So pyruvate dehydrogenase is an enzyme that requires a lot of coenzymes. One of them is thiamine, which is a vitamin, but in the form of thiamine pyrophosphate, which is what we see here as TPP. So we have lipoic, which is another coenzyme obtained from lipoic acid or lipoic acid. whatever you want to call it. But it is also very important in the formation of acetyl-CoA, especially in the access of pyruvate to all of these subunit active sites in the pyruvate dehydrogenase enzyme. We also have flavin adenine dinucleotide, which is represented as FAD, and its major function as a coenzyme is to support lipoid. So it's like lipoid works a lot and then could get tired. So FAD is there to help it, to support it, to be like a supporting arm to it. So that is it for this major reaction. So at the end of the day, all of these interactions make acetyl-CoA, which is the entrance enzyme, like the ticket to enter into this cycle. And since this enzyme is very, very important, it is no surprise that deficiency of the enzyme will lead to very severe disorders or syndrome. And in most biochemical pathways, when you have specific enzyme deficiencies, they lead to very severe metabolic diseases or syndromes. And the one we have here is the Wernicke-Korsakoff syndrome. Syndromes usually are a combination of different symptoms that aid or indicate a particular disorder. But it's mostly genetic or very chronic disorders that you see syndromes. Syndromes, most organs, not just one organ maybe is affected, most organs are involved. But in this Wernicke-Korsakoff syndrome, you have metabolic acidosis. I think you can infer from the name. Metabolic, which means it's related to metabolism. Acidosis means increase in acid. So the blood becomes acidic because of increase in metabolites that are acidic. And metabolic acidosis can lead to coma or death eventually. So this syndrome is also associated with the Wernicke encephalopathy and Korsakoff psychosis. So Wernicke encephalopathy is basically characterized by confusion, loss of muscle coordination, loss of vision, all of that because of the accumulation of these acids in the brain. All of these would affect different parts of the brain that are in charge of muscle coordination, in charge of vision and all of that. And then the psychosis is also associated with hallucination most times. So this is an overview of the TCA cycle. It looks like a roundabout where we have the entrance molecule coming in from a certain direction, acetyl-CoA. We are going to see that some of these intermediaries that can be gotten from other metabolic pathways, some of the intermediates can also go into other metabolic pathways. So going into the step-by-step process, the most important thing for you to know is to know what happens at each step so that you can maybe probably guess the enzyme and also the product. So the first step is citrate formation. So you're forming citrate from what? The molecules that enter the cycle. What do we say enters the cycle? Acetyl-CoA, which is a 2-carbon molecule, oxaloacetate, which is a 4-carbon molecule. So you can guess how many number of carbons citrate has. So citrate would most obviously have 6 carbon molecules. And what is the chemical nature of citrate? If you have been following the class keenly, I mentioned it already. And another hint is that it is linked to the name of the cycle. Okay, what is its chemical nature? Well, tell me in the comments. Second step is an isomerization step. In one of the first lessons of introducing carbohydrates, I think we talked about isomers and what does it mean. Molecules that have the same chemical formula but maybe differ in their structure. So if citrate has 6 carbons, it's obvious, and this is an isomerization, it's obvious that isocitrate will also have 6 carbons. So the enzyme here is aconitase. You would notice that the cell is very intentional in what they do. You might be asking, why does it have to interchange it? Then in chemistry, you would see that in this isomerization, there's breaking of bonds and forming of new bonds. So probably for isocitrate to be able to react to the next enzyme to produce a product that is necessary, citrate has to have a certain bond somewhere. And that's what the body tries to do. The cells, they are so smart. Cells know that we have to change this one to look like this in order for this one to bind to it or to react with it, something like that. Now we come to the third step, which is oxidative decarboxylation. So there is an oxidation that happens here, and there is a decarboxylation. From the name, decarboxylation, D means removal or reduction. Carboxy is carbon dioxide. So in this step, the carbon dioxide leaves. So you can guess that if carbon dioxide is leaving, 1 carbon is leaving from the molecule. And so the product here has 1 carbon less than the reactant. So alpha ketobutyrate has 5 carbons, while isocitrate has 6 carbons, because there is a decarboxylation. And the enzyme also gives a hint about this oxidation. Oxidation is usually addition of oxygen or removal of hydrogen. That's why you have a dehydrogenase, it's removing hydrogen. So basically, it answers the question, why is it decarboxylation termed oxidative? Because in addition to decarboxylation, there is an oxidation happening to isocitrate. The next step is also another oxidative decarboxylation. So in this case, 5-carbon alpha ketoglutarate will be reduced to succinyl-CoA, which is 4 carbons. Now we come to another interesting step, and this step you have succinyl-CoA converting to succinate. Here you think this is an isomerization step. They have the same 4 carbons, but there's a difference. And if there's a difference, why is this step not called isomerization? I'll give you some 2 minutes or 1 minute to think. You can pause the video and think about it and try to answer, but I'm going to answer it for you. Here, the step is a substrate level phosphorylation. So there's something else happening within this reaction that is not just an isomerization. So basically, in a reaction termed a substrate level phosphorylation reaction, an energy molecule is produced. We know the energy currency to be ATP. And if the energy currency is ATP, it means that even if you have other currencies like GTP, it's another energy molecule, it has to be converted to ATP for the body to use it. So in this particular reaction, an ATP molecule or an energy molecule is produced. That's why we call it substrate level phosphorylation. So it's basically the molecule was existing. So either ADP was already existing, and then they just added a phosphate to make it ATP. So in this case, GDP existed, and then you just added a phosphate to it to make it GTP. So in substrate level phosphorylation, you're adding a phosphate to an intermediate of the energy molecule to produce the molecule. So you're not producing the energy molecule from scratch. That's why you call it substrate level phosphorylation. As opposed to when the ATP molecule has to be produced from scratch in the electron transport chain. So I don't know if you understand. Let me know in the comments if you need further explanations. So basically in this step, an energy molecule is produced. That's why you call it the substrate level phosphorylation. And the energy molecules are phosphates. That's why it's phosphorylation. To be able to make ATP without going through the electron transport chain, the reaction that makes that ATP or GTP is a phosphorylation reaction, substrate level phosphorylation reaction. All right, the next is another oxidation, but in this case, there's no decarboxylation, so we have 4 carbons. So we can, since it's a cycle, you can begin to guess that we're getting closer to the end, because the starting molecule is oxaloacetate, which is 4 carbons, and acetyl, which is 2 carbons. If we have come to 4 carbons, maybe we're going back towards the beginning. So succinate that was obtained from the substrate level phosphorylation is converted to fumarate, which is another 4 carbon molecule. But here it is not just rearrangement of bonds. There is actually a dehydrogenase that occurs. So it's not an isomerization reaction. So it's an oxidation reaction. And the next one, fumarate is converted to malate through a hydration. Hydration, when you're hydrating, basic terms, you're drinking water. So in this reaction, water is also entering to make this malate. And finally, malate is converted to oxaloacetate, which is one of our starting molecules. So the procedure here is also a dehydrogenase enzyme that is involved. Definitely, hydrogen is removed or oxygen is added, which is an oxidation reaction. So basically, that is it for the Krebs cycle or the TCA cycle. As we said, we're going to come back to the energetics of the cycle. So, as we said at the beginning of the lesson, the major aim of the TCA cycle is to produce molecules or energy molecules that are going into the electron transport chain to produce ATP. And we are going to see the electron transport chain in the next lesson. The molecules that go into this chain to produce energy are NADH and FADH2. NADH is nicotinamide adenine dinucleotide, and the H there just means that it is a reduced NAD or reduced NADH. And then we have FAD, reduced FAD when the H is present. So it is flavin adenine dinucleotide. So these 2 molecules are what carry or go into the electron transport chain to result in production of the energy currency that we know, which is ATP. So in this TCA cycle, I want you to write in the comment section how many NAD are produced and how many FAD produced. In the electron transport chain, we're going to see how many ATP molecules does one FAD produce and how many ATP molecules does one NAD produce in order to know their impact in energy production. So you have the number of NAD that are produced in this cycle and the number of FAD in the comment section. We also see the production of a direct energy molecule, GTP. Although the body does not use energy as GTP, GTP can be directly converted to ATP. So 1 GTP is equal to 1 ATP. So the body has still gained or produced energy through the cycle. So at the end of the day, you should be able to calculate the number of ATP molecules that will be eventually produced by 1 TCA cycle. And remember, you'll be able to do this when we talk about the value of NADH in terms of ATP and the value of FADH in terms of ATP when we look at the electron transport chain. Now, for every cycle or every metabolic pathway that we have seen, we have looked at regulation. And we already know by now that regulation is always at the level of some key enzymes. Some enzymes that are very important, not like the others are not, but those enzymes are the ones that if there is a deficiency, the cycle may not continue. So we have the first one is pyruvate dehydrogenase complex. Remember, it's a complex of 3 subunits, and I mentioned already how important that particular enzyme is. We also have citrate synthase, have isocitrate dehydrogenase, have alpha ketoglutarate dehydrogenase. So citrate synthase, from the name, it's the enzyme that catalyzes the synthesis of citrate. Isocitrate dehydrogenase is the one that catalyzes the first oxidative decarboxylation of citrate into alpha ketoglutarate. And then you have the alpha ketoglutarate, which is the next oxidative decarboxylation enzyme, And so all of these enzymes can be inhibited by certain molecules or activated by certain molecules. In the case of an inhibition, if you're inhibiting these molecules, it means you already have enough energy in the body. You don't want any further production of energy. And so if you have enough energy, there's excess ATP. Also, if there's excess citrate, it means a lot of citrate is being produced, and also eventually a lot of energy is produced. Excess NAD also implies that a lot of energy is present. Excess succinyl-CoA also would inhibit this pathway. And activation is done by almost opposite molecules. So if there is high levels of ATP and AMP, which are intermediates of energy, then it's showing that the body does not have enough ATP. It instead has a lot of the intermediates and need to be processed into ATP. So all of these are going to activate these enzymes or bind to these enzymes and tell the enzymes that you should continue the cycle. All right. So in addition to feedback inhibition that occurs within the cell by itself when there is excess ATP, when there's excess ATP or ATP intermediate, there is also inhibition that can be done by exogenous substances out of the body. Something like fluoroacetate is found naturally in some plants as sodium fluoroacetate, and I'm going to show you an example of the plants that are common around Australia, Africa, and Brazil, for example. Also, this fluoroacetate is then used to produce pesticides that are used against plants or rodents. This inhibitor is a suicide inhibitor. Suicide inhibition means that it completely stops the enzyme from binding, and so the pathway does not continue. Suicide, from the name, the cell will die because it cannot produce energy for itself. Arsenide is another inhibitor which is commonly found in groundwater, and it's usually due to industrial processes that release this chemical into groundwater that contaminate it. So basically, arsenide is a non-competitive inhibitor. A non-competitive inhibitor is an inhibitor that binds another space on the enzyme. It inhibits alpha ketoglutarate in a non-competitive manner, which means that the enzyme's substrate can still bind, but the inhibitor also binds in another site, and it can also change the complete structure of the complex that is formed and cause it not to function optimally. So malonate is another one that is found in some fruits and vegetables, and could also be found in groundwater due to industrial contamination. But in the fruits like pineapples, which are especially in green pineapples that are unripe, you would find malonate in them in very little concentrations. But if you consume them in higher concentrations, or malonate is found in these in higher concentrations, it will probably competitively inhibit succinate dehydrogenase. And competitive inhibition means that it competes with the substrate to bind at the enzyme active site. So it's not going somewhere else like arsenide to bind. It wants to enter the same place where the substrate is supposed to bind. And so if it is in higher concentration, it's going to overtake. Another interesting fact to know about the TCA cycle, it's very central. And one of the reasons is because it's an amphibolic cycle. Amphibolic means it has both anabolic reactions and catabolic reactions. This table gives a summary of the end of the reactions that are anabolic TCA cycle, because we already know that all the other reactions of the cycle are catabolic. They are breaking down molecules to produce energy, while the anabolic ones involve mostly synthesis or reaction or molecules that are going to go into other reactions to produce other molecules. So that's what we're going to see here. Here we have pyruvate producing oxaloacetate, have citrate producing acetyl-CoA, producing malonyl-CoA. This is important in fatty acid biosynthesis. Have alpha ketoglutarate converting to glutamate, which is involved in purine or important in purine synthesis. Purines are bases that are necessary in forming nucleic acids. We're going to see it later. We have succinyl-CoA, which is an intermediate that adds to glycine to produce alanine, and it is important in heme synthesis. Heme is very important part of the structure of hemoglobin, and know that hemoglobin is the important unit of red blood cells. Oxaloacetate is also converted to aspartate, which is important in pyrimidine synthesis. So the nucleotides that we know, or the nucleic acids, the bases, are either purines or pyrimidines. So we see that the TCA cycle is a very busy junction, like the roundabout that is always very busy. So the molecules, the intermediates that are produced are always used up continuously. So they are basically supposed to be used for energy or other intermediate production. As we have seen, we have metabolic reactions that are obtained from some of those molecules. Even the molecules in the catabolic cycle can also be used for continuous production of energy or even other intermediates. And so there should be a replenishing source for some of these molecules, and that's what we call anaplerotic reactions. Reactions that replenish the intermediates that are produced in or that are needed in the TCA cycle. So if oxaloacetate is limited, pyruvate carboxylase would help to produce more of it by converting pyruvate to oxaloacetate in this reaction that adds carbon dioxide and ATP to it. Also, if malic is limited, pyruvate can still come into action through the malic enzyme to produce malate. The same as aspartate can produce oxaloacetate and glutamate can produce alpha-ketoglutarate. These are done by transaminase enzymes. These enzymes, from the name transaminase, simply transfers an amino group from one molecule to another. And in the process of transferring, they produce new molecules too. And so if there is limited oxaloacetate or limited alpha-ketoglutarate, these enzymes are going to replenish them. Basically, these are the basic things that you need to know about the TCA cycle. As you said, it's a very busy roundabout. It's a very important and central metabolic pathway. That's why we have seen a lot more about it. I hope this video was helpful for you. I hope you were able to grasp one or two things. And if you have questions, as usual, you should write in the comment section. And I'm also waiting for your responses to the questions that have been asked. I'm also waiting and expecting you to like the video, to share it with other people that you think this might be of help to, and also to subscribe, please, so that in the future people can be able to find these videos.